This video demonstrates the recommended post-delivery handling and culture workflow for the ready-to-use organoids. The workflow includes package inspection, medium preparation, organoid transfer, and culture and maintenance. Check the package and cryovials for damage upon receipt. Gently tilt the tube, allow the organoids to settle at the bottom, and check that they remain intact with no disintegration. Before starting the experiment, pre-warm the organoid maintenance medium to 37 degrees Celsius for 15 minutes as specified in the product datasheet. Using a cup-tip Pasteur pipette or a wide-bore pipette, gently remove the ready-to-use organoids from the cryovial and transfer them into an ultra-low attachment 6-well plate. Remove the shipping medium. Add pre-warmed maintenance medium to 5 milliliters per well, making sure the organoids are fully submerged to prevent exposure to air and drying. Place the 6-well plate on an orbital shaker at 100 revolutions per minute and culture for 48 hours at 37 degrees Celsius with 5% carbon dioxide. Replace the maintenance medium completely after 48 hours. Keep the organoids at 37 degrees Celsius and 5% carbon dioxide with a full medium change every 2 days, and regularly monitor organoid morphology and overall culture condition. Note: when changing medium, slowly add pre-warmed medium down the side of the well to minimize shear stress and prevent mechanical disruption of the organoids. The organoids are now ready for the further experiments. Best of luck with the study. Please contact Acro Technical Support with any questions.